Hello, everybody. I am Pablo, supervised by Mark Blackster. My pronouns are he, him, they, them. I'm gonna talk about programmed chromosome breaks in Oscheius nematodes and how these impact large-scale rearrangements. So first of all, nematodes, they have very divergent genomes. As you have seen here, at the bottom of this plot, you see different reference organisms compared to different relatives. You can see that C. brixi and C. plicata, which are both Caenorhabditis, are almost as different between each other as Drosophila melanogaster from brine shrimp. That is to say, they are very different, even though they look very similar, just little worms. When you go and look at their chromosome composition, actually they're quite stable. You can see that there are 7 ancestral chromosomes, which we inferred from comparing these chromosome level assemblies. And we found that each of these ancestral units is identified as a currently extant chromosome in at least 2 independent branches of the tree. The most outstanding exception is for ascaridomorph, which has many chromosomes, but this is a phenomenon of which I will tell you more about later on. So to better study chromosome evolution, we did what we would do, sequence more genomes. We sequenced more Oscheius genomes, and to our surprise, we found that they have different number of chromosomes, which is totally unexpected. We expected to see 6, and instead we found some with 6, some with 8, 13, and 16. For those of you who haven't seen a high C, each red block represents 1 chromosome. Now, to understand this, we need to learn about programmed DNA elimination, abbreviated as PDE. PDE was discovered in nematodes more than a century ago, and it consists briefly of 2 steps. Here we're seeing the early stage embryo, 4 cells. These are germline cells, so they will keep the whole genome and unbroken chromosomes, but somatic cells will undergo the process of programmed DNA elimination. This involves double-strand breaks, and this will happen when chromosomes are in the metaphase, and then the fragments that will be retained, those will be the new chromosomes. The DNA that is left at the mid will be later on sequestered and eliminated. So that's how you go from having a whole chromosome to having more but fewer, more but smaller chromosomes. Now, it's also important to highlight what did we sequence. We sequenced a lot of nematodes from different stages, and different stages have different proportion of germlines. So altogether, we have a varying proportion of germline in our sequencing datasets. When Oscheius tipulae, the first Oscheius we sequenced, we found that every single chromosome end removes DNA from their ends. And we know that because here we see the alignment of long reads. These are uniquely mapping, so we know they can only be placed here. And some of them have telomeres, which are, they are just telomere. It extends all the way, just telomeres. So the only place in the genome where you find that is at the very chromosome ends, nowhere else. So we know these are chromosome ends in some cells, the somatic cells presumably, and some other sequences continue going without telomere here. So we presume that these are the germline-restricted sequences. Taking into account that, then we can put together these different shapes into 6 germline chromosomes. Some of these could be easily assembled thanks to removing only a few base pairs, and we having enough coverage of long reads to cross over and capture non-telomeric reads supporting this junction. We also asked ourselves, is there any common sequence at these break sites, or how would the cell know where to break? So we extracted every sequence where the DNA is predicted to break via telomere addition. And we found that the same motif is found across different Oscheius at these positions. So you would expect the telomere will be added either in this direction or in this direction. So the break will be happening around here. So not only that, we also found that some break sites are nested. In this region, you found telomeric reeds supporting the break at this position, and this position, and this position, and this. If all somatic cells were breaking the DNA exclusively here and here, we wouldn't have enough coverage to detect telomeric reeds mapping at these positions. So that indicates that different somatic cells are removing only this part, and other somatic cells are removing, in addition, this and this part together. Additionally, we found that programmed DNA elimination regions have a surprising correlation at the boundaries of inversions. When we compare the gene order between different Oscheius, in this case, such as the Lecura JU1382, we found that they co-occur at the breaking synteny. Also, in some cases, these break sites separate by ancestry. This purple tend to be the ancestral X, while the green indicates an ancestral autosome. So in some cases, programmed DNA elimination is separating between ancestry of the chromosome. In summary, we found that all ends are remodeled. Some chromosomes have internal scissions. Some of these remove hundreds of base pairs, and they contain satellite repeats and genes. Other breaks remove just a few base pairs, that is, no genes or repeats are removed. And we also found one case of a nested break site, which indicates that DNA elimination has been co-opted to subdifferentiate somatic cells. Also, all break sites share a common sequence motif. And additionally, break sites are associated with inversions. I would like to thank this great team of scientists, without them, it wouldn't have been possible to find this, especially my supervisor, Max Blackster, and Alan Tracy, who has a lot of decades of expertise in analyzing genome. Thank you all for your attention.